Hello everyone, my name is Maria and today we're interviewing Professor Jess Frith from the Department of Material Science and Engineering. Her background is in stem cell biology and bioengineering. Welcome today.
Thanks, nice to see you.
Okay, so we'll start off with, could you tell us a bit about your academic and professional journey?
Okay, yeah, so if I start way back at school, I was always interested in sciences, so I took all the opportunities I could to study biology, chemistry, a little bit of maths. I actually don't have a formal engineering background. So when I went to uni, I studied biology, and that was a whole mix of learning about plants and animals and all sorts of different things. But as I got further through, I was more and more interested in our bodies and how they repair themselves and what we can do to promote that. And so then when I learned about stem cells, I was like, well, that's fascinating because the potential we have to heal our bodies and repair them with stem cells is immense. So then after my first degree, I tried to avoid PhD actually. So I did a master's in biomolecular science. But then I realized that if you want to do research and if you're interested in that side of things, PhD is the way to go. So I did a PhD focusing on stem cells, but already the first hints of engineering are creeping in because the problem I was tackling is how are we supposed to culture these cells so that they behave like they do in our bodies. And so I was developing a way to culture the cells in 3 dimensions rather than as a flat sheet on plastic, which now I think sounds obvious, but back at the time was quite new, and we had to convince a lot of people that was a sensible idea. So all of that was in the UK at the University of York. And then after PhD, I thought that I would move to the other side of the world. So I moved to Australia for a postdoc, and that's when I first joined proper engineering faculties. So I was at the Australian Institute of Bioengineering and Nanotechnology, and that's where I learnt all of the engineering, all about biomaterials, functionalizing surfaces and lots of different things, and saw how we can use those to work with stem cells. And then after being there for 5 years, I got an ARC DECRA fellowship, and with that I moved down to Monash and I set up my group here at Monash.
Amazing, that's a really interesting trajectory. We'll get more into that later. Could you share a bit about your current research and its potential impact in regenerative medicine mainly?
Sure. So the core of our research is thinking about how cells sense the physical environment around them, and then thinking about how that affects what they do and how we might use that to repair body tissues. So one side of our research is very fundamental, and we use biomaterials to try and present really specific environments to the cells so we can gain fundamental understanding of how they respond to different cues. Then the idea is that for the applied part of our research, we've learned some of those things that help us design scaffolds to put into the body or better ways to grow the cells for cell therapy, and so we can apply our findings to do things that will actually help people.
Amazing. So you mentioned that your work bridges stem cell biology and engineering. How did you navigate the challenges of working across disciplines?
Yeah, look, so it's not easy, but I think the trade-off is that if you mix the disciplines, that's where it's really interesting. So there was a steep learning curve. I had the strong background in biology from everything I studied, where it was hard. When I first joined an engineering group, it was hard. There was different terminology to learn, different ways of thinking, perhaps. Biology often, it doesn't work in straight lines. It's so complex, it often doesn't do what we expect. And I think from an engineering perspective, we're used to designing things and we know how they'll respond. So bringing those kind of concepts together. But look, I think it's valuable because once you put that hard work in, it opens up a lot more avenues.
Yeah. And I can imagine that it would also be useful for more people across many different disciplines. So
yeah, it broadens what you can do. Yeah. I think the key thing for me is the realization you don't have to know every little bit about everything. but you have to know enough to realise who you need to work with and what help you need. And then it's about developing, the skills to collaborate and work well with other people.
Okay. And regarding the experience in the UK and then more recently in Australia, how have all these environments influenced your research approach?
Yeah, interesting question. I guess one side of it is, I think it's important to have exposure to different places. Part of that could be UK versus Australia, but part of it as well is, engineering versus a biology lab, or even, how our team works versus the group next door to us. Everybody works a bit differently.
That's true.
So rather than there being necessarily a big difference between the UK and here, I think just seeing and observing the different ways that people work and figuring out what suits you is a good thing to do.
Yeah, for sure. So now we'll get more into stem cell and tissue engineering questions for those students that are interested in this field. So my first question was, we know that mesenchymal stem cells are widely studied, but what do you think are the biggest challenges in harnessing them for clinical applications?
So all of those challenges, I think, actually, in essence, come back to engineering. One problem we have with MSCs is that there's not actually a really good description of what they are. Mm-hmm. So we can get them from lots of different places in the body. We can get them from bone marrow, we can get them from umbilical cord, and each of these sources of cells is slightly different, but technically they're all still an MSC. So that creates complexity, but then also my MSCs will be different to your MSCs, so we've got variability within different people. So when you're trying to design a therapy, one of the big challenges is having a consistent product or consistent cells at the end so that we know how they're going to behave.
Okay, and for a bit of context, how do mechanical cues influence MSC behavior?
Okay, so that's a big question. So MSCs are adherent cells, which means that in the body they attach. So they attach to the extracellular matrix in our tissues. So one of the ways they do that is by the different matrix that's around them. So they can sense the biochemistry of what's around them. But also when they attach to things, they generate force and they pull against that matrix. And depending on how that deforms or moves and various things like that, there's a lot of information that comes through that gives the cell information on how stiff or soft that material is and lots of things.
Interesting. And so some students might know you from the biomaterials unit. So what role do biomaterials play in enhancing these MSC therapies?
Yeah, so look, they're critical. They're critical for the fundamental understanding because we need to design materials where we can control things like the stiffness or what we present to the cells. So that's one aspect. Then if we want to grow good quality stem cells that we can put back into patients, we need to think about what materials we use to do that. And then the third thing is what materials do we use to deliver the cells back into the body. So if I'm going to try and engineer a tissue, I'll typically need some kind of material scaffold to provide the structure and framework for that tissue. And there's lots of detail about the structure of that and the shape and the pores and different things that will influence how well we manage to build those body tissues.
Sure. And could you talk a bit more about the application that this could have on, for example, musculoskeletal injuries or degenerative diseases? How could they help?
So look, I think one example is a project that we're working on with a neurosurgeon at Monash Health, so just next to campus. And I guess we all know people that have a dodgy back. In many cases, that's due to the discs between the vertebrae. And so it's really common that they just degenerate or degrade as we get older, and eventually that causes pain. So the surgeon we're working with, he basically does surgeries where he tries to sort of reinforce those discs. And at the moment, we can't really do it very well, and ultimately we often end up removing them and just putting lots of metal in and sort of reinforcing everything. But we're working with him to basically create a sponge-like material, and we've put a drug inside it, and then we put our stem cells on it, and he will put that inside the disc, and the drug helps the cells regrow the tissue, and the sponge provides a way for us to deliver that drug slowly, but it also provides the framework for that new tissue. So hopefully, if somebody has, discs in their back that are really dodgy, they don't end up just having lots of metal, but we can regrow the actual disc.
Yeah. That's so cool. It's so fascinating. So moving on to the future of regenerative medicine, where do you see it heading, and what advancements do you think are necessary for it to really push forward?
Yeah, so look, the regenerative medicine in its truest form is still in very early stages, I would say. And I think, so the first real treatments are just starting to come through. One of the things we've learnt is our stem cells modulate the immune system and help use the immune system to promote tissue repair. So I think that's one of the big things that we will do. But I think really, the crux of making everything happen is we've got to work out how do we grow lots of these cells that are still really potent and do the things that we want. How do we get them to the right place in the body and stay alive? And then it's the tricks of how do we better get them form the tissues that we want. And those things seem quite trivial, but we still don't actually have a good answer for a lot of those.
Yeah, I can imagine. I mean, something that I thought of when you were talking about the discs is once, that has regenerated, how do we know that it's not going to, dissolve or disintegrate again in the future, given that it would most likely be an elderly person? I guess stem cells could also help maybe with that, the aging part of...
Yeah. So I guess one of the opportunities is, because our stem cells age with us, right? And they get less able to repair our tissues. Yeah. So I think one of the opportunities is to be able to use someone else's cells. And that's one of the attractive things about MSCs, because they're somewhat protected from our immune system in that you can use cells from one person in another and they're quite well tolerated. Something else that's very exciting, we've been working with a company called Cynata Therapeutics. So they make mesenchymal stromal cells from induced pluripotent or iPS cells. Yeah. And that gives a sort of universal donor that you could use for a lot of people.
Wow.
So that type of research is pretty exciting.
Okay, very cool. Are there any other emerging techs or trends that have particularly excited you?
So, well, one of the things our group is really interested in at the moment is about the cell-cell communication. And we're more and more thinking that the way the cells, talk to each other is the key to getting that tissue repair to happen. So we're looking more and more about the factors that cells release in different environments. So one challenge is we're trying to put our stem cells into a tissue that's degraded or diseased, but that makes them behave differently. Yeah. So how do they then talk to the other cells around them to repair that tissue? And I think if we understand that cell communication better, then we'll be able to engineer ways to make the cells talk in a way that promotes them all to repair that tissue.
For sure, yeah. Now we'll move on to our second last part, which is about teaching and advice for students. So for those students interested in stem cell research and who are listening to this and are thinking, wow, this is all so cool, what skills or areas of study would you recommend they focus on in their degree?
Look, to a certain extent, I guess you need to formally study, the different underpinning disciplines. So try and get exposure to the biomaterials, to the cell biology, to the devices and the bioengineering. But the other part is actually more just in developing your curiosity. So take the time to think about things, take the opportunity to talk to your lecturers, to the researchers. There's a lot of people around that know a lot of things and are very happy to sort of talk and provide advice. So I would say, aside from the formal study, make the most of the opportunities around to learn about what's going on around you.
Yeah, I think most students would be surprised at how many lecturers are willing to just have a chat with you and have their door open. So, yeah,
for sure.
Good advice. If you could give one other piece of advice to students pursuing biomedical engineering, and you know, thinking back to when you were a student, what would it be?
Yeah, I think similar to what I said, like have confidence in yourself, have the confidence to ask questions. Like, people always say there's no such thing as a stupid question. That's pretty much true. And just immerse yourself in it, ask lots of questions. I think that's the best thing you can do.
Yeah, very good. So now we're going to finish off with our personal and fun questions. If you could work on any futuristic bioengineering project with unlimited funding, what would it be? That's tricky. Well, with unlimited money, one of the things I'm really interested in is looking, I guess, at the cells, and we now have the ability to, with omics technology, to get readouts of every single gene that every cell expresses, every single protein that cell is making, even like metabolic things those cells are making. I would love to combine all of that with sort of AI, machine learning, computational technology, and look at, as we build synthetic tissues, what's happening to every single cell in a tissue to help us work out how do they sense what's around them, how does that affect them, how does that affect their neighbours, and how does this all come together as a network to make the big tissue or organ. I mean, it's very interesting that you say this, because there might be students who are more interested in the AI aspect, but now they might be listening to you and thinking that they could use those skills in this area as well. So it's good you've brought it up.
Yeah.
Now, do you have, I guess we've touched on this a little, but do you have a favorite scientific discovery or also scientist or experiment that has influenced your career? This is always a tricky one.
It is a tricky one. Well, I guess two things come to mind. One is the discovery of PCR, so polymerase chain reaction. I think from a being in the lab perspective, like that's something that absolutely changed our ability to analyze what's happening. And there's a nice story about it, about the guy having a dream and it came to him like he was surfing on a wave or something.
I think I've heard that,
But another experiment that I always think about and I think is very elegant is one that was done to definitively show, and these were blood stem cells, to show that they were a stem cell. And there's an example where they isolated these hematopoietic stem cells from an animal, and they repopulated the bone marrow of another one, and it all grew back. And then they did it again, and you could keep doing it. And the fact that you can keep doing it is the thing that tells you you've got a true stem cell in there.
Oh, very cool. Now, if you hadn't been a bioengineer, biologist, or a researcher, what career would you have pursued?
So, my other career plan when I was about 15 is very different. So I used to play the trombone.
Okay.
And I wasn't bad. And so my alternate career is that I was going to try and be like a professional musician and play for like the Royal Philharmonic Orchestra or something.
This question is always so funny. Every person just says something completely different. Okay, anyways, moving on. So then finally, outside of research and teaching, what are some other interests or hobbies that students might not know about?
I guess I like to try and keep active. So I play a bit of soccer when I'm not at work.
Do you have a team that you play with?
Yep, so our team is called the Penguins, and we're part of the Aspendale Stingrays Club.
Okay.
So you might find me on a Sunday afternoon out playing seven-a-side soccer.
Very cool. And no more trombone then?
I still have it,
Yeah.
but I think for the sake of the neighbours I haven't played it for a while.
Okay. All right, well, thank you, Jess, so much for this interview. I hope that now students feel a bit, that they know you a bit better and they feel comfortable approaching you after class if they want to keep talking to you about all things MSCs.
Definitely, always happy to answer questions.
Great, thank you.